So predicting how changing just one tiny part of a protein, a mutation, affects what it does, that's like a really core challenge in biology, right? And biotech, too.
Absolutely, it's critical. Think about understanding diseases or designing better enzymes, better medicines. It all comes back to this.
And there's been this massive push using, well, really complex computer tools,
It,
deep learning, protein language models, all that stuff.
yeah, exactly. The really cutting-edge computational methods.
Right. And you kind of think, okay, more complex equals better predictions, more data, bigger models, should just work perfectly.
That's the expectation. But the sources we looked at point out some issues, like these big models can be black boxes. You don't really know why they're making a prediction.
Ah, the interpretability problem.
Exactly. And surprisingly, their accuracy in predicting mutation effects.
Yeah.
It isn't always, let's say, head and shoulders above everything else, despite all that complexity.
Which leads us to the main point from these sources, I guess, this like almost shockingly simple model.
Precisely. A model that seems to perform right up there with, and sometimes even better than, these sophisticated state-of-the-art approaches.
Okay, so let's get into it. This model is called RSALOR, and our mission today for this deep dive is really to understand why something so simple works so well, at least according to the sources.
Okay, so RSALOR. It's basically built on just two main ideas, both pretty grounded in biology. First, evolution, looking at how conserved a protein part is.
Cisluridation.
And second, structure specifically, how exposed or buried that part is. Relative solvent accessibility.
Okay, let's take the evolution part first. Conservation. That feels intuitive, right?
Yeah.
If an amino acid at some spot has stayed the same across loads of species for ages, evolution's basically screaming, don't touch this, so changing it is probably bad news.
That's the core idea. RSALOR uses something called a log-odd ratio, or LAR score. It gets this from looking at how sequences of related proteins line up.
A multiple sequence alignment.
Right. And the LOR score basically asks, based on evolution, how likely is the new amino acid compared to the original one at this position? If you swap something common for something really rare at a spot that hardly ever changes.
The LR score flags it as potentially disruptive.
Exactly. It captures that evolutionary signal.
Got it. Okay, evolutionary pressure in LOR. Now, the structural bit, relative solvent accessibility, RSA.
Yeah, RSA. It's just a measure of how much of an amino acid is sort of sticking out into the water versus being tucked away inside the protein's core.
Buried versus exposed.
Uh-huh. And the general rule is that messing with residues buried deep inside usually causes more problems, destabilizes the protein more than changing ones on the surface. The core is packed really tightly, very specific interactions.
Okay, so RSALOR combines the LOR score and this RSA measure. How does it actually, like, mathematically put them together?
Again, it's really quite simple. It takes the LOR score, the evolution part, and it scales it down based on how exposed the residue is.
Scales it down if it's exposed.
Right. It uses 1 minus the RSA value. So if a residue is totally buried, RSA is near 0, the scaling factor is near 1, the LOR score basically keeps its full weight.
Okay.
But if it's fully exposed, RSA is near 1, the scaling factor is near 0, so the LOR score's impact gets minimized.
Wow. That is really straightforward.
It is, and the sources argue that's why it works. It captures this basic biological logic. The really bad mutations are likely the ones that are evolutionarily unlikely and happen in a critical buried spot.
Right.
Simple, but it's the key features.
So the big question, does this simple logic actually hold up when you test it on real mutation data? How well does it perform?
Well, according to the sources, yeah, pretty impressively. They tested it on a big benchmark called Protein Gym. Has about 2.5 million mutations, measuring effects on stability, activity, things like that.
And the result?
RSALOR got an average Spearman correlation of 0.473 across all those data sets.
Okay, 0.473 Spearman correlation. Put that in context for us. Is that good? How does it stack up?
Spearman correlation looks at ranking, does the model rank mutations from bad to okay, similarly to how experiments rank them. Closer to 1 is better. Now, 0.473 isn't perfect, obviously, but the sources report it's right in line with, or even slightly better than, most of the complex methods they benchmarked it against.
Even the big protein language models?
Yeah, most of them. The sources noted only one specific approach, combining structural info with language models, did marginally better overall in their tests.
Okay, this is where it gets really interesting for me. A simple model performing this well. What does that imply?
It definitely raises questions, like what are these super complex models actually learning? Maybe the simple fundamental features, conservation, burial, are doing a lot of the heavy lifting anyway? Or maybe the complex models aren't using these basic principles as effectively as they could? It also just highlights that predicting these mutation effects accurately across the board is still, a really tough problem for any method right now.
And practically speaking, RSALOR has some big advantages too, based on the sources.
Oh yeah, big time. It's apparently super easy to use. Just a Python package, one command to install, no training needed, no fine-tuning.
No training. That's huge.
Right. And it's fast. The source mentioned it chewed through all 2.5 million mutations in that benchmark in less than 20 minutes on a regular laptop.
Wow. Fast, simple, easy. Any downsides? Limitations mentioned?
Yeah, the sources acknowledge it's simplified. It assumes, for instance, that mutations on the surface, 100% RSA, have zero effect,
Mhm.
which isn't always true.
Mm.
And it completely ignores epistasis, how mutations interact with each other.
Right. Context matters. But even ignoring those complexities.
It still performs surprisingly well,
Yeah.
which really underlines how important those basic evolutionary and structural factors are for single mutations. And here's one last really cool finding from the sources. They actually checked if adding RSA information could help other models.
Oh, what did they find?
They found that adding RSA, just that simple structural feature, significantly boosted the performance of many other models in the benchmark, even some complex ones that already use some form of structural data.
Okay, so wrapping this up for you, our listener, it seems like maybe integrating these straightforward, interpretable features like how buried a residue is, how conserved it is, could be a really key step, not just for simple models, but potentially for making even the most advanced AI tools in protein science more accurate, maybe more reliable, perhaps even easier to trust. Definitely something to chew on as you think about these tools.